Drug development process prepared by Ruhul Amin. For M-Pharm clinical research subject. Introduction. New drugs have revolutionized the practice of medicine and support healthcare in various demographic outbreak and managing disease. Example as deaths from cardiovascular disease, the main cause of death in the USA, and from stroke have decreased by more than 50% in the USA over the past 30 years. Increased use of antihypertensives, cholesterol synthesis inhibitors, drugs that prevent or dissolve blood clots, medical devices, and drug-releasing stents. First steps in the development of a new drug is the discovery or synthesis of a potential new drug molecule. Understanding of its interaction, mechanism, with the appropriate biologic targets. Repeated application of this approach leads to compounds with increased potency and selectivity. Assessment of the relative safety in animals. With regulatory approval, human testing can then go forward in 3 phases. A 4th phase of data gathering and safety monitoring is becoming increasingly important and follows after approval for general use. Cost of development of a single new drug that reaches the marketplace estimates from $150 to $900 million. Only 3 of 10 marketed drugs return their research and development (R&D) investments, thus providing considerable motivation to develop blockbusters. Thousands of compounds may be synthesized in hundreds of thousands tested from libraries of compounds for each successful new drug lead. During the second half of the 20th century, estimates indicate that medications produced by the pharmaceutical industry saved more than 1.5 million lives and $140 billion in the costs of treatment for TB, poliomyelitis, CAD, and cerebrovascular disease alone. New drugs played a key role in the post-1995 decline in HIV mortality, and the social returns for HIV drug innovation appear to be extremely large. Approaches to drug discovery. This approach is known as classical pharmacology, forward pharmacology, or phenotypic drug discovery. The various approaches to drug discovery include: Step 1. 1. Identify and define the medical need. 2. Research on disease. 3. Search for lead components. 4. Optimized the properties of lead components. 5. Performed drug development and preclinical study. Step 2. Preclinical trial. Step 3. Clinical trial. Step 4. Post-marketing surveillance. Identification of novel targets. Novel receptor and enzyme genes have been cloned and expressed from human genomic libraries using the techniques of molecular biology. Identification of the function of a newly identified gene potential target for drug screening. Genomic target identification. Identification genomic targets. Expression profiling, comparison of CNS derived from normal and diseased tissues using hybridization. Positional cloning, gene mapping in combination with population genetics involved in human diseases. Search of lead. Most new drugs are discovered or developed through one or more of 6 approaches. 1. Identification or elucidation of a new drug target. 2. Rational drug design of a new drug based on an understanding of biologic mechanisms, drug receptor structure, and drug structure. 3. Chemical modification of a known molecule. The 6 approaches continue. 4. Screening for biologic activity of large numbers of natural products, banks of previously discovered chemical entities, and large libraries of peptides, nucleic acids, and other organic molecules. 5. Biotechnology and cloning using genes to produce peptides and proteins. Efforts continue to focus on the discovery of new targets and approaches, from studies with genomics, proteomics, nucleic acids, and molecular pharmacology for drug therapy. 6. Combinations of known drugs to obtain additive or synergistic effects or a repositioning of a known drug for a new therapeutic use. Target optimization. A biomolecular target, most commonly a protein or a nucleic acid, is a key molecule involved in a particular metabolic or signaling pathway that is associated with a specific disease condition or pathology or to the infectivity or survival of a microbial pathogen. Potential drug targets are not necessarily disease-causing but must by definition be disease-modifying. Study of target, its characteristics in terms of interactions with new compounds. Ligand site topology and functional effects, cloning. Available to facilitate high-throughput screening. Functional assessment of the activity of new compounds. Thank you for watching video. Continue in next video.